Okay guys, so I said I'd make up a quick review for you guys for your test on Friday. So what I'm going to do is go through our genetics unit and talk about the different types of crosses that we've had, that we've talked about in class. And when we go, we're going to be looking at college prep. You guys are going to be looking at these 5 crosses: monohybrid, incomplete, codominant, sex-linked, multiple alleles. Those are the 5 that you have to focus on. But honors, when you see at the end there the dihybrids, that's for you honors guys. College prep, you guys can just skip over that one of the last ones that we do. I believe I have it in pink. So let's get started with our first type. We got monohybrid crosses here, and we'll say, let's go back to Mendel and look at his plants. We have tall plants and short plants. Well, in a monohybrid cross, we would take, let's say we know the, let's define our alleles. Our alleles would be, and we'll say that tall is dominant. So since tall is dominant, we use T as our letter, and we'll say that we're going to cross 2 tall plants and see what happens. For this, we're going to say that we have 2 heterozygous tall plants. So we defined our alleles as T, and that's the dominant. We are going to our next step, which is defining the parents, which is this here, our heterozygous tall plant with a heterozygous tall plant. So next step is to actually do our cross. Set it up here. Make our box. And then bring each allele down in order to see what our offspring, what the probability of our offspring would look like. So in this case, we have our 2 plants that we crossed here and here. We end up with 25% chance that the offspring will be homozygous dominant, 25% chance that our offspring will be homozygous recessive, which ends up being this short guy over here, and then a 50% chance because it's both of these 2 boxes. Let me change the color on here for you. These 2 boxes both represent a heterozygous tall plant. But if we're talking about how many tall plants in general, we would say that we have a 75% chance for them to be tall, because all 3 of these are tall. Because if we remember, the dominant trait shows itself, so therefore any one that starts with our capital T would end up being tall. So we'd have 75% chance of the offspring being tall and a 25% chance of them being homozygous recessive and short. Now let's put the ratios on here, our genotype and phenotype. So our genotype would have 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. And our phenotypes would either be tall or short, and that's a ratio of 3 to 1. Let's move on to our next set of crosses, and we'll start with incomplete. And remember, there's 3 possible outcomes for both incomplete and codominant problems, but incomplete is where we have a mixing or a blending of our characteristic or trait. So in this example, I have 2 animals, 2 cats, either a red cat or a blue cat. And these 2 are going to cross in order to find out what the offspring would look like. So if we're going to define our alleles, let's just say, and when we give you problems, we would normally define them. So let's define that right now. We're going to define our alleles as, we'll have R being red and lowercase r being blue. But if you remember, I said that there's three possible outcomes. So therefore, in this case, we'd have to define our alleles as capital R, and lowercase r for capital R red, lowercase r blue. And then our third one would be incomplete, and when you mix these two together, we have purple. So in this example, we can go ahead and set up our cross that we originally had said, and I said, so we defined our alleles now, we have all three possible types, and we also have what I said was going to be our parent cross, which is the red with the blue. So now we'll set up our cross, and we have our parent goes on either side, and then we just do our cross like we normally would. The big R's come down and the little R's go across. So in this example, if we look at our genotypic and phenotypic ratios, we have all four genotypes being big R, little r. So therefore, our offspring, if we look back up here, we see that that would end up being 4 purple cats. Now for this example, we're going to look at codominance, and if you remember, I said that we can have 3 phenotypes in this one as well. But with codominance, our notation's a little bit different, and we're going to define our alleles first. So this guy up top, these are horses, so we'll define this as horse. He's got a black skin color, and then the one on the bottom is horse white color. So these are our 2 parents that we're going to cross. When we cross these, put this up in the box up here, so we know that these 2 are going to be crossed. We have our black horse up top and our white horse on this side, and we do our cross just like every other one. Bring the B down, the W over, and this goes for every single box here as well. This is all going to end up having HB and HW in every single box. But what does that actually mean? So we said we have HB, HW as the offspring. Now with codominance, neither one of these is dominant over the other. They're both dominant. That's the codominant. They're both dominant. So that's why we have the big letters for black and white. So in these problems, if it's not given to you that it's a codominant problem, the key terms that we talked about were spots, stripes. So in this case, all of our offspring would end up being a mixture of the 2. So let's first write our genotypes. Would all be HB, HW. We'd have 4 of those. And our phenotypes, well, I didn't show you one yet, but they would all end up being, I'm just going to call it zebra, because they'd be white and black striped. So we'd have 4 of those, and that's where we get the mixing, or the, I'm sorry, not the mixing, the stripes. We'd have stripes, and it'd be black and white striped animal. So those are the key terms that you're looking for when you're trying to find out what type of problem we're looking at. Our 4th type of cross is sex linkage. And we said in class that there's 3, in terms of our classroom, we have 3 different key terms to look for. And that's color blindness, hemophilia, and boys versus girls. So anytime you see that, you automatically know it's going to be a sex-linked problem. So let's do the first one here. We're going to focus on colorblind. So let's go ahead and define our alleles. Let's say our problem is we have a mother that is, I said mother, so we know we have two X's, right? We have a mother that is heterozygous for being colorblind with a father that is normal. So our normal, we're defining our alleles. We have XN being normal, and when we see X little n, or X little n Y, we have colorblind. So those are our alleles that we're going to be using. So what I did was I defined our alleles here. We have XN being normal, X little n, or X little n, Y. Those are colorblind, and I just went ahead and set up the cross for you guys. This was our second step, our defining what our parents are going to be. So we have our normal vision dad at the top here, and our heterozygous mom right there. So I'm just going to go ahead and do the cross, and we bring our X down from dad and our X over from mom. Here we bring our X over from mom and our Y down, and then again we have dad's N coming down, X, and mom's X coming across, mom's X coming across, and dad's Y coming down. So in this example, our genotypes, we have 4 of them. We have one female that is homozygous dominant for normal vision. We have one female that's like mom and has the dominant allele, as well as the recessive allele. We have one that is like dad, has normal vision, and we have one male that is colorblind. So that's our notation, and then I just described to you. We have our phenotypes end up being, get a different color here. We have 2 normal females, 1 normal male, and 1 colorblind male. For our college prep guys, this is the last one we're going to go over. So in this, for multiple alleles, we said we're going to be talking about blood typing. So in this case, we have dad being type AB and mom being type O. So if we define our alleles here, we have IA, IB is type AB, and little I is type O. So if we go ahead and we have our parents defined now, so this is what our cross is going to look like. So we'll go ahead and set that up. Change it to red because we're dealing with blood. Go to put dad up top, IB, and mom is little I, type O blood. So our offspring are going to end up getting 1 from each parent. The B is coming down from dad. The little I is coming across from mom. So in this case, we only have 2 options. We have type A blood and type B blood. And we have 2 of each. So if we wrote that out in genotypic ratio, that is that. And then if we wrote out the phenotype, we just write 2 type A and 2 type B. Now, because you can only have a 15-minute video, I'm going to have to switch over to do the honors dihybrid on another video, as well as the extra problems that you guys are going to need for remediation.
Made with Doodlecast Pro.